Can you see the screen?
Not yet. Is sharing.
Are you able to see now?
No, sir.
Oh.
I think you close and restart if possible.
Reshare, sir.
Just let me try again.
Close that presentation.
Is it there now?
Yes, sir.
Yes.
Okay, good.
Stop कर दो.
So shall we start?
Yes, sir. You can start.
Okay, thank you. Good afternoon, everyone. Today we will discuss about resting state fMRI, and we will also touch upon voxel-based morphometry. And voxel-based morphometry is a technique where you can compare two brains regarding their white matter, gray matter, and other constituents. The difference in the brain as far as white matter and gray matter are concerned. You can find out whether due to aging or due to training, the gray matter or the white matter is increasing or decreasing in the brain, depending on what you plan you are experimenting. So first we will cover the resting state fMRI, and then we'll go on spend some time on voxel-based morphometry, because both the techniques are very good when you're studying the brain and trying to look at the cognitive profile of different disease conditions or different disorders, including aging. And also you can also see the beneficial effects of meditation, yoga, and also the training. If you're giving some training to some person or acquiring a second language or third language, you can see how the brain reorganizes itself in terms of gray matter and white matter concentrations and resting state connectivity, how it changes after the training. So we discussed last time about the bold contrast, which is blood oxygen level dependent contrast, and this exploits the properties of magnetic properties of blood. And we discussed last time, when blood is oxygenated, it is diamagnetic. When it is deoxygenated, it is paramagnetic. And paramagnetism, does a lot of dephasing on the signal, so signal is reduced. So the basic concept behind bold fMRI is, whenever you have any sensory, motor, or cognitive task, suppose I am moving my finger, my motor area in the brain is active, it will get more blood in that area, more oxygenated blood. So because of this oxygenated blood, there will be a deoxyhemoglobin ratio will be decreased, and we'll have less spin dephasing, and there will be increased signal in that region. So particularly only in that region there will be increase in MR signal, which is picked up by NMR, by MRI, and you can play around with that signal and generate the functional maps. So we also discussed that because the change in the signal amplitude is just 2 to 5%, which is as good as noise in MRI. So in a typical MRI experiment, fMRI experiment, you have to acquire thousands of images in two different conditions. One is a task condition, one is a resting condition. And when you do an average of these two conditions, subtract the two images, then you can see the pixels which have seen the oxygen levels, and you can then superimpose this on high resolution MRI and find out what are the areas which have seen the change in oxygen. And you can see here, these are the areas which are shown in color. They are seen, in all these areas there is a change in oxygenation, and you can see these areas which are responsible for this particular task. So this is a basic technique how you do a task-based fMRI. And like similar way, if you do experiment, eyes open and eyes closed, you can see, and this is a resting when eyes are closed. When eyes are open, this is the signal changes. You can see how the signal changes from eyes closed to eyes open, and then you can see these are 3 to 5% signal changes. And after you process the signal, you open minus close, you can see the region of occipital cortex, which is responsible for the vision, You can see the brain activity in that region and find out the area which is responsible for the vision. Okay, similar way you can perform any experiment depending on the what is the research question. If you want to look at the areas of working memory, attention, or decision making, you can design a task paradigm in such a way that invokes that particular function. So this task paradigm is itself a very complex area, and most of the psychologists can do this for you, who can design a task depending on the cognitive parameter you want to study. So as I said, the signal changes between normal oxygen levels and elevated oxygen levels is just 2 to 5%, and it is as good as noise. So what we do in resting fMRI, we ask the patient to lie down in the magnet, and we don't ask him to do any task. So that's why it's called resting state fMRI. Here no task is done, and we want to look at the brain, how brain is utilizing its energy when the person is at rest, when he's not doing any goal oriented task. When we are talking, we are writing, we are studying, we are reading, we are doing a goal oriented task. But when the person is lying quiet, he is not doing any goal oriented task, so he's in random thoughts. So we are trying to map the brain when person is in a random thought condition. So resting state fMRI is like task-based gold fMRI only, and measurement requires exactly as task-based fMRI. You have to use a gradient echo pulse sequence or the EPI pulse sequence. Actually, it is exactly the same experiment, but you are not performing. The subject is not performing any directed mental task. That is the only difference between task-based fMRI and resting state fMRI. And then you are trying to study a time course of spontaneous bold response measured. No, you can see here there's no activity. You can see there's only noise in this signal. This experiment, there's only noise. So we are only trying to study this noise, how this noise is varying in different regions of the brain. And we know that in the computer networks which ping together, they are connected. Same way here, we are trying to see the different areas in the brain which are in synchronous activity, where the noise is in synchronous with other areas. So we assume those areas are connected to each other, and then you can compute the networks in the brain which are effective at one particular moment. So, and the advantage of resting state fMRI is it is much less demanding than the task-based fMRI, because there's no task. You don't have to design any task, and many times the children and people who are sick, they are not able to adhere to the task. They are not able to perform the task properly. So it becomes very difficult to perform such experiments when there's a kid or the people who are not able to do the task properly. And this gives you a very good in a snapshot. It gives you a good idea about the functional connectivity in the brain when the brain is not doing any mental task, directed mental task. So what is the motivation behind studying noise in bold signal? We know the human brain is 2% of total body mass, but consumes about 20% of the total energy, most to support ongoing neuronal signaling. So that means when the brain consumes almost 20% of the total energy of the body, looking at the weight of the brain, which is much less than the body, but it consumes 20% of the total energy. Then second thing is, task driven, when you're doing any task driven job, then only 5% energy is being utilized. So what is happening to the rest of the 95% energy? So this is where we feel, scientists felt that 95% of the energy is being utilized when you are not doing any goal oriented task. You are in random thoughts. So we always talk there's a lot of junk going on in our brain when we are not doing anything. So we are just thinking about various things, various possibilities about our past, about our future. So we are using all this energy into these, unnecessary junk ideas. So what we do, and if you have to see where this energy is being utilized, how these networks are working when the brain is, not doing any very significant task. You can study this using resting state fMRI. So the first experiment was done by the Biswal, an Indian scientist, who took the area in the motor cortex, the right and the left motor cortex, and studied the noise. See the correlation between the noise from the two hemispheres, the left motor area and the right motor area, and also the supplementary motor area, which I showed you last time. He found a very good correlation between the two areas, and our correlation coefficient was almost 0.84, which is a very high correlation between left motor area and the right motor area. Whereas when he did a correlation between the motor area and the visual areas, he saw there was no correlation between these two areas. Correlation was only 0.06, which is almost no correlation. So he thought, when you are getting a correlation between the areas which are related to each other, which are linked to each other, so we should be able to generate the network maps using this board signal. And so now then neuroscientists started playing with the spontaneous board signal to learn about the intrinsic functional connectivity of the brain. And in the initial stage, Fox et al. In 2007, he identified many of the networks, which is called primary motor area, primary visual area, extrastrial area, insular temporal area, various resting state networks they identified, and they published this in 2007. Similarly, later, many more scientists also worked on this, and the question was, why to study brain at rest was, because we know there are localized functions in the brain, like motor area is very localized, but they are also distributed in the brain. So we wanted to study localization versus connectivity. All these different areas in the brain are connected to each other through the neural networks. And understanding the intrinsic organization of the brain to establish clinical cognitive biomarkers. And if you have a disease case, you can find out how in a disease case, these resting state networks, the connectivity gets affected. Like if you are studying Alzheimer's, you can see how the memory network gets affected in Alzheimer's, and then you can also find some markers which can tell you, okay, because of this network, the connectivity issues in this network, there could be a possibly, cognitive decline and the, respective disease. One can have, one can establish clinical or the cognitive biomarker of a particular neuro disorder. Can be done on any population without much ease and relatively less setup and expertise. In task-based fMRI, you also need very expensive, accessories, as I showed you last time. This is a projector, projection device, which itself is 1 crore, and then you have to design the task, and then take the psychological battery. So it becomes very complex. So here you are not doing anything; you are just doing the resting fMRI. You're just acquiring the signal while the person is at rest, and you can using fMRI, resting fMRI, you can distinguish any two populations on the basis of their functional connectivity. You can have a healthy, population, and then take any disease condition, second population, and see how these networks are affected in a disease state. So very, very versatile technique, and you can see the number of publications in this area are, increasing exponentially, because more and more people are shifting from task-based fMRI to resting state fMRI. So these are some of the more networks which were identified later: language network, default mode network, somatomotor network, visual network, and so many networks. And people are still till now they have already identified about 17 to 18 resting state networks in brain, and people are trying to identify more and more networks which are pre-existing in the brain, and more and more work is going on. So these are another publication where you have default mode network, executive control network, executive network right and left hemisphere, then salience network, then sensory motor network, auditory network, visual medial network, visual lateral network, visual occipital network, cerebellum. All these networks have been identified by people who are working in this area, and it is it is a great thing that with a snapshot you are able to see all these networks and see which all areas, which all networks are affected because of disease process or because of, or which networks can be strengthened by giving training, acquisition of second language, third language, or training somebody on meditation, yoga, or anything where you can train a person, maybe in physics or maybe in computational, maybe in computer programming. One can look at these, how these functional networks, they vary when you give a training to a particular person. So the most important network which we see in the brain is default mode network. Default mode network has several components, and I don't know, but I will like to mention here. There are about 6 areas which form the part of default mode network, which is posterior cingulate cortex, anterior cingulate cortex, medial prefrontal cortex, precuneus, inferior parietal cortex, and hippocampus. These are the 6 areas which constitute the default mode network. And default mode network is called task negative or intrinsic network. That means if you ask a person to do any task, the default mode network shut down. It shuts down. Because now since you have to do a goal-oriented task, now you can no more do a random thinking because your brain gets engaged to do that particular task. So default mode network is the only network which is task negative. That means it will get deactivated when a person is doing any task specific job. And it is known to be involved in mind wandering when our mind is going here and there. Retrieval of autobiographical episodic memory. We are thinking about our past or our future. Then self-referential mental processing. Within ourselves, we are thinking how I should plan my future, on envisaging the future and considering the past, how what others are thinking about me, what I think about others. This all is contained in default mode network. And this default mode network is also people who are negative, who have negative thoughts, people who have positive thoughts. They also are contained in this default mode network. So a very, very, very important network, which is and we have seen in most of the disease conditions, default mode network gets affected, and there have been changes. People are seeing changes in the connectivity of subregions of DMN, and associated with number of psychological disorders such as schizophrenia, epilepsy, anxiety, depression, autism, ADHD, and Alzheimer's disease. There are several publications which have come out, and they have also found that in both autism and depression, there's a failure to deactivate DMN during goal-directed tasks. That means people who have depression and autism, they are not able to shut down their DMN, so that they are not able to do their task properly. Whenever they are given any goal-oriented task, they are not able to do it effectively because they are not able to disengage their DMN, which has to get disengaged to do any particular task. So DMN can be potential biomarker for monitoring the therapeutic response of various interventions, including meditation. Suppose you have a person who has a working memory deficit, and then you do interventions on him. Whether it is a drug-related intervention or it is meditation or psychotherapy, whatever you do. So then you come back, see how these networks are improved after the person has been given intervention, therapeutic. You can monitor the therapeutic response using resting fMRI. You can find what are the networks which are affected, and then when you give intervention, how these networks are getting corrected, their connectivity patterns improve after the intervention is given. One can really have a look at this. So it is a very powerful technique to study the monitor the therapeutic response of various interventions. And at the moment, we are also at TDO where I am working now. We are trying to see the role of Brahmi Grita. Brahmi is a herb. We are trying to see how Brahmi Grita can be used in older population and show how this improves the cognitive performance in elderly population more than 65 or 70 years of age. We are monitoring them before we give them intervention, and then we are giving them Brahmi Grita for 3 to 6 months, and then get them again for resting fMRI and MRI to see how these networks have improved in functioning. And then we can also correlate these functional connectivity with the parameters, the psychological battery, like if you are doing a working memory or attention or decision making or anything you want, long-term memory, short-term memory, and then the quality of life, quality of sleep. All these parameters can be studied and then correlated with the functional connectivity in the brain, and also see how default mode network is functioning much better than what it was functioning earlier. In case there's any question, please you can interrupt me.
Hello, participants. If any questions, you can ask, sir. Don't think.
So second very important network is the
Sir,
salience.
1 minute, sir.
See, Professor Khusu, sir, I just want to know, see, when people dream, see,
Yeah.
so which part of the this default mode network is at that time, does it,
Actually, there's a
what
very
is the dream state
very-
of brain?
this is a very interesting and very intriguing question you ask. Dream doing dream studies in fMRI, I don't think people have tried, but people have tried with the EEG. They have, because when the person is sleeping, you can fit the EEG cap on the patient and monitor the changes happening during the night. So this will be very, very, very important, very, very interesting to see what happens to the different networks in the brain. But it will also definitely, as I said, even during dream also, default mode network will be activated. 100% there will be activity in the default mode network also. But how the activity will be shown as compared to the when a person is awake, that is to be seen. How the activity patterns will be different when a person is asleep, in dream, or in when he's in awake condition. So this is a very interesting thing. I think people will definitely try this when you have more more robust experiments to plan, and when you can put the person in the magnet for the night, let him sleep, and then monitor this continuously. So this is a very important question, but I don't think anybody has done the resting fMRI during dreams.
Okay, okay, sir, thank you, sir.
So then next important network is the salience network. It has two areas: dorso-anterior cingulate cortex and fronto-insular cortex. In short, it serves to identify the most relevant stimuli for guiding behavior in response to several internal or external stimuli. Basically, when suppose you are sitting idle, you are not doing anything. I tell you, okay, recite a shloka. Now you have to go from random thought to a goal-oriented task. Now this salience network, what it will do, it will give you inputs that you have to shut down the DMN and start a relevant network, relevant areas. You have to perform this task now. It will send internal signals that this DMN should be disengaged now, because there's a task which is waiting to be done. So basically, salience network, it detects the stimulus, and then the frontoinsular cortex initiates the appropriate transient signal to engage the central executive network to mediate attention, working memory, and all other higher cognitive processes. Because now if you have to recite a shloka, you have to activate your auditory cortex. You have to do the memory task. You have to make a decision when to start. So all these networks will be signaled, and also at the same time it will disengage the default mode network, because default mode network has to get disengaged, because you can no more afford to have random thoughts now, because you have a very task oriented goal oriented task now. So this is what the salience network does. Actually, we don't realize how much is happening in our brain. We take everything for granted, but how signals are going, and then they're putting you into attention, and asking to start a job, which whatever has been given to you. So this switching mechanism helps to focus attention on external stimulus and eventually allow this stimulus to take added significance or saliency. That means now this job which has been given to you, it takes the priority over the default mode network, and you start performing that task, in a goal-oriented manner. So this is what the salience network does. Then, then the next important network is central executive network, which is dorsolateral prefrontal cortex and postparietal cortex. And this network plays a main role in active maintenance and manipulation of information in working memory and in judgment and decision making in the context of goal-oriented behavior. When you have to do a goal-oriented task, the central executive network comes into action, and it sends signal to different regions of the brain. Okay, be ready for this particular task. Just like like when you have some project comes, or you have to give some job to your team, you call them. Okay, now this is the task what we are going for next 1 week. So similar way, this central executive network also tells the different regions. It computes what are the regions required for that particular task, and then sends a signal on those areas, so the task can be completed successfully. Then is the frontoparietal control network. It has two regions, lateral prefrontal cortex and inferior parental cortex. This is purely for decision making. So when you make decisions about anything, the frontoparietal control networks comes into play. So this becomes really active when you have to make a decision. Suppose I tell you to compute 4 plus 6 plus 9 plus 10. So you will compute, and then when you have to make decision, when you have to speak out, take a decision, okay, this is the value, then frontoparietal control network will activate the auditory cortex and you will speak out. So this is how the brain is organized within itself, and then how different signaling processes take place from one part of the brain to another. Then there are more, and all these networks which I am discussing now, they are all task positive networks. That means when you give this task to a person, there's an increase in activity. DMN was a task negative network. When you give any task, DMN gets deactivated. But all these networks get positively activated when we call them task positive extrinsic networks. The next network is cinguloocular network, which is medial superior frontal cortex, anterior insula, and anterior prefrontal cortex. It is involved in performing goal-oriented tasks. So whenever you have any goal-oriented task, it will be taking over. And suppose you are given 2-3 tasks, so you should also be able to prioritize your task in such a way that the most important task is done before you do a less important task. So this is all done in the cinguloocular network. Then sensory networks and motor network. When you do any motor task or sensory 5 senses, do any any sensory jobs you do, then somotomotor sensory motor cortex of the network gets activated. And when you are hearing anything, then it is the auditory network, which is facial gyrus, superior temporal gyrus, and posterior insula. These are the 3 areas involved in auditory network. Then comes the attention network. You have dorsal attention network. Then you have in the attention network, you have 2, dorsal attention and the ventral attention. In dorsal attention, you have interparietal sulci and frontal eye field. This is involved in the execution of control of attention. And ventral attention network is involved in the detection of salient cues, those stand out from the environment. That means these are the cues when you take from environment. Suppose you are looking at some scenery, you are taking cues from that scenery while making any decisions in your task. So this is ventral attention network, which is actually responsible for the detection of salient cues. Then are the language networks. We know Broca's and Wernicke's areas are the regions where we have which are involved in speech production and articulation. So these are the language networks. Then we have visual network, which is occipital cortex, which I said is responsible for the vision. Whether you have, and this also has different regions within visual cortex, which gives you color discrimination and all that. Is all stored in visual networks. So these are the networks which you are able to see through resting fMRI, and you can make judgment which all networks are affected in a particular disease condition. So, and then you can also look at the psychological battery and try to correlate the functional connectivity with the psychological parameters and see whether these parameters are they really correlating, or you are getting a false signal. So when you do resting fMRI, we discussed last time, the processing, the pre-processing of the data is exactly same as you do in task-based fMRI. And then after you do this, then you do a lot of noise regression, because this is a you are only studying noise, and you have to see the correlation between noise in 2 pixels. So the lot of you use this individual component analysis, ICAs, and then try to see what is noise and what is signal, and then try to filter out the noise completely. And then you in the first go you may see about 100 networks, but they are lot of noisy networks. So slowly, when you denoise your data, slowly you will come down to 6, 8, or 10 networks which are actually activated at that particular point of time. So this is how you can clean your data to show the resting state networks in a healthy and in a disease population. So this is a resting state fMRI in hypothyroidism. In hypothyroidism, people have attention deficit and also motor deficit. So if you have to do this study these hypothyroids using task-based fMRI, you have to do a separate fMRI for motor and then separate fMRI for your attention. So here, when you do resting fMRI, you are able to see there are 4 networks which have been visualized here, 5 networks. And then so you can see these are the networks: right frontoparietal attention network, left frontoparietal attention network, somatomotor network, default mode network, and medial visual network. So these are 5 networks which are found in these 2 cases. When you do the comparison, then you find in the functional connectivity is diminished in hypothyroid subjects as far as medial visual network is concerned and motor networks are concerned. These are the 2 regions where they have found reduced connectivity in hypothyroid subjects. So in one single shot, you are able to see what are the attention, what are the these cognitive decline or the cognitive impairment in these hypothyroid subjects in one go. Similarly, then you can also look at the different regions and see how much is the connectivity differences, left hemisphere, right hemisphere. This all you can also quantify your connectivity data by doing quantification of these signals. This is a resting state if I'm in schizophrenic patients. You can see these 6 networks were identified in them, and then after you do a analysis of the further analysis, do compare the 2 populations, you find left frontoparietal network, lateral visual network, medial visual network, and motor network and DMN, which are affected in you can see DMN is present in all the disease conditions because they also have negative thoughts. Schizophrenics also get negative thoughts, and they have hallucinations, they have visual hallucinations as well as audio hallucinations. So they are quite a bit, their DMN is affected quite a bit, and I also told you they are not able to shut down their DMN when they are giving a goal-oriented task. So this all can be seen by using this technique of resting fMRI. So this resting fMRI has been, people have tried in different disease conditions. Somebody who's interested can look at the literature. They have tried in pre-surgical planning and rehearsals, Alzheimer's disease and dementia, disorders of consciousness, neuropsychiatric and neurodevelopmental disorders, neuroendocrine disorders, in stroke, in traumatic brain injuries, in anxiety, depression, PTSD, and also in environmental stresses like high altitude, thermal, and radiation stress. Any of these areas, people have tried to see how the brain is getting affected by various stresses or by various disease conditions or disorders. And people are also trying to see how these networks can be strengthened by the help of training or meditation or interventions, and how these networks can be improved. The connectivity can be improved in disease conditions by treating the disease through interventions. So all this is a very hot topic nowadays, and a lot of people are working in this area. particularly people who are interested in neuroscience. A lot of neuroscience research is happening, and also people from psychology, and social sciences have also tried to, they also trying to fit in their questions into this field very aggressively. Then after this, I will go for a second technique, which is called voxel-based morphometry. And voxel-based morphometry is a whole brain unbiased automated technique that examines the local composition of brain tissue on a voxel to voxel basis using structural magnetic resonance imaging. We discuss what is T1 weighted image. We discussed this in our second lecture. And T1 weighted images, I also told you, T1 weighted images are very good for anatomical details. T2 weighted images are very good for pathological details. So here in voxel-based morphometry, you acquire 3D images or 3D T1 images of the brain with very high resolution, as good as 1 millimeter slice thickness, and also 1 by 1 millimeter box, these pixel dimensions. So then you try to compare, if you want to compare two populations, you will compare two brain images voxel by voxel. You will compare and then see whether the voxel fills into the gray matter area or the white matter area or CSF or whatever, So this is basically a automated technique where two populations can be compared using we call it MP-range. MP-range is a sequence which is magnetization prepared rapid acquisition with gradient recall. So these are very fast technique. It takes about 6-7 minutes to take the image of the whole brain, and then one can do the processing offline later. And it provides a faster and more comprehensive analysis of the changes in brain structure. Now we know that we always say that person has more gray matter or less gray matter, and we know that with aging and by disease process, there's a changes in the gray matter and white matter concentration in brain. And this is a very beautiful technique where you can actually look at the brain volumes and gray matter volumes in different structures, the different structures in the brain, and also the total intracranial volume and gray matter and the white matter, and then compare this with another population which may be diseased or which may be trained for a very specific acquiring new skills. So this is another good technique. And VABM is sensitive to minor differences in gray and white matter after global difference in anatomy and position has been discounted. And it's not easy because when you're comparing two brains, the brain size and everything is different. So you have to do a lot of processing on the data to make them on the same template so that you can compare these two brains easily. Bear with me, this slide has got stuck. It will take some time to move further. In the meantime, if you have any questions or any clarifications, you can ask.
Yeah, participants. The big file, sir, or
It is this slide is okay.
it has started? Yeah, it has come.
It has come. So when you have these high resolution images, then you are acquiring, say suppose you are acquiring 10 healthy or 20 healthy brains. Then we all know that all these 10 subjects will have different brain size, okay, head sizes. So they need to be normalized to a standard space, which is a standard template, so that all brains can be translated into this template so that they look alike. So the size can be made similar. Then after you do this spatial normalization, then you do a segmentation of the tissue classes. Then you segment the brain into gray matter, white matter. These are algorithms which are utilized to see only gray matter, to see only white matter, or to only see fluids or the CSF in the brain. Then you also, since you are now normalized the images, you will adjust for the volume changes during normalization, because you are, some places you are expanding the brain, sometimes some places you are compressing the brain so that they fit into the standard template. Then you do modulation, which adjusts for total volume changes during normalization. Then you smoothen the data. Each voxel is weighted every surrounding voxels. Smoothing improves the signal to noise ratio. What we are trying to do is, by smoothing, we are trying to improve the signal to noise ratio of these images, and then you perform the statistics by comparing the two populations and make the inferences about the differences in these the two segments. So this is a normal the scheme. You have a original image which is here, then you do a normalization with this standard template. This can be template, actually mostly people are using the Western population template. Now India also, people are trying to develop our own template, because our head sizes are much smaller than the people abroad. So because their total body structure is much bigger than us. So, but you can also use this average image of 100, 200 patients, 200 volunteers, and then make a standard template yourself. So what you are doing, this is the original image of the patient, then this is a standard template, then you do a normalization. After you do normalization, you see a picture which is normalized. Okay. After you have a normalized picture, then you send it for a segmentation. You have GM, gray matter, template, white matter template, and CSF template. Then you do a this segmentation, and you fire gray matter segment, gray matter segment, and then smoothing gray matter. And similarly, you have white matter segment, the modulated white matter, and the smoothen white matter segment. And similarly, you will have 3 CSF segments as well. So you will generate these images by doing this process, and then finally you are putting a Gaussian filter to smoothen the data, and then you have finally this smoothened image, which can be now, which is ready for comparing between the two populations. So the normalization process is aligning images by warping to standard stereotactic space, and it has, as I told you last time in functional MRI, brain is a 3D object, and you can take it as a solid structure, which can be defined by translational and rotational parameters. And then depending on the brain size, you can do scaling, sharing, so that you can have you can compare, you can fit this into this template. So all these things, all these processes have to be carried out so that it fits into this template. Then you take the second brain and then do the normalization of that brain, and take one by one and put them together. So this is what you do. This I have shown before also. This is the original image. This is a template. Then do spatial normalization, and then you have a spatially normalized image as shown here. So this is a process which is called spatial normalization. It's basically to account for the different size and shapes of the head of different people. This is the pre-processing step to do voxel-based morphometry. Then is this segmentation. You have a brain image, then you segment this in gray matter, white matter, and CSF. So here you are seeing a white matter structures more prominently. Here you are seeing gray matter structures, and you are seeing only CSF, which is becoming bright. Wherever there is a fluid, you have a signal here. Rest are all dark. Rest have been suppressed. So this is only showing a CSF map of the brain. So now you have 3 images: gray matter, white matter, and CSF. Then you do a volume modulation, because when you do normalization, I told you you are playing with the images. So you have to do this modulation so that all the effects of total volume, what was present in the brain earlier, you have to maintain that and try to do a image volume modulation, and create an image which is modulated, gray matter of modulated white matter and moderated CSF image. Then you do a smoothing, and smoothing is done like this. You can see this is a gray matter image. You can see how it is done with this smoothing with a 8 millimeter kernel. Generally, this FWHM can be taken as 2 to 3 times of the slice thickness, sometimes even more. So you can see it has been smoothened now. You can see all the variations which are there in the image, unsmoothed image, are gone now, because we have much better signal noise ratio to compare the two subjects. Then you do a probability maps. You do this statistical analysis to see whether a voxel which you have selected, does it fall into the gray matter intensity, white matter intensity, or the CSF intensities. So this is what you do by spatial prior probability maps, and then keep on counting these gray matter voxels and white matter voxels, and then find two different populations. So what you do, you create this. I told you earlier, we create T1 weighted MRI from one or more groups at a single time point. Then analysis, you compare whole or part of brain volume between groups, or correlate volume with another measurement at that time point. So you can see, you can also do the internal structures. You can look at the volume of the hippocampus, volume of the frontal cortex, volume of the hypothalamus. You can calculate all these volumes within the brain separately, and then see which areas in the brain have shrinked because of aging or because of the disease process, and then how the gray matter and white matter has changed due to the disease or aging or any external stimulus. So this all can be, so basically a question in this is, when you have two populations, suppose you have Alzheimer's, and then the control population you have subjects there, and then you take a single voxel and ask, are the intensities in the EAD images significantly different to those in the control images for this particular voxel? And is the GM intensity, is this a GM intensity lower in the AD group of controls? And then do a two sample t-test, see whether these differences which you are seeing, are they significantly different or they are similar. So this is how you will compare the two brains, voxel by voxel. And then you do it from for all the voxels, and highlight all voxels where intensities are significantly different between groups. So then you can show them in the form of, like, here this is the result, how you show these are the areas where the gray matter volumes are different from the healthy controls. So they are lower than the healthy controls. So you can see these are the areas. Then you can also have look at the stereotaxic atlas and see what are the areas, what are the broad main areas, and then also determine the structures, whether it's hippocampal area or the gray, or the hypothalamus or frontal lobe or parietal lobe or occipital lobe, whatever, depending on the location and the coordinates, you can find out which area of this of the brain belongs to this particular clusters. This is an alcoholics. This study from our center, where we wanted to study the people who are with alcoholic disorder, and we can show then. And this is a glass brain view of the brain, which shows all the regions as a see-through view of the brain. Wherever you are seen any volumetric or the gray white matter changes, these are shown in the pictures over here. And this is a how the area in the these this white matter loss and gray matter loss is there in alcoholics compared to healthy controls, and you can show them on the brain surface. This is a 3D rendered view of the brain surface. I can show these areas and how volume and densities, gray matter, white matter densities have varied over the due to the alcoholism or any other disease process. So this is loss in gray matter. In alcoholic dependent, significant gray matter volume loss was observed bilaterally in putamen, in inferior frontal gyrus, postcentral gyrus. These are the areas where you have found the reduced gray matter volume. And similarly, you also have a loss in white matter and in different regions. And in alcoholics, we have found most of the losses were found in the right hemisphere and not much areas of loss were found in the left hemisphere. That is probably the reason in alcoholics you don't find any language issues. You see a alcoholic person, he will lose all his senses, but his language skills don't get affected so easily. They are still able to talk and speak whatever they want to speak. But other areas like attention, memory, balance, everything is disturbed except the language, probably because there's no area in the left hemisphere which is affected in alcoholism. When we saw the this VBM analysis. Similarly to resting fMRI, people have tried applications in all the type of disease processes: schizophrenia, autism, bipolar disease, temporal lobe epilepsy, Down syndrome, Alzheimer's, Parkinsonism, aphasia, healthy subjects. And then you can also do this in healthy subjects to see how the gray matter volume increases or how the connectivity increases, because now you are becoming, when you give training, you become more smarter. So that means your gray matter and also the connectivity between the different regions is improving. So you are able to do tasks in much better way. Then acquisition of new skills, second level acquisition, and also you can also study the aging, normal and pathological aging, and how these parameters vary when a person ages. So this is a very important project what we are doing at the moment at TDU, working on the intervention of herbal interventions to improve the cognitive profile in elderly population. So I think I have finished my talk today. If you have any questions, please raise them.
Yeah. Hello, participants. Any question you can ask now? Sir, before that, I have a small question.
Yes, please.
Sir, what is the compositional difference between this gray matter and white matter?
See, there are a lot of differences between the two, issues. This gray matter. Actually, I can tell you briefly. You have seen circuit boards, no? Electronic circuit boards. Boards have components,
Yeah.
which are doing local functions. Then all these circuit board, these are connected. These components are connected by tracks on the circuit board, right? So similarly, the gray matter is the areas where these functions take place, and white matter carries the signals from one place to another. They connect the neurons from one place to another. They transmit signal from one place to another.
Okay.
So they have very definitive roles, the gray matter and the white matter. So white matter are like connecting wires. So when we look at DTI, diffusion tensor imaging, I will show you how these tracks can be seen using diffusion tensor imaging. And you can see how these tracks go from different orientations. And this is that gives you a connectivity which is fixed connectivity. They are actually connected with those tracks in the brain. We call this white matter fiber tracks. That is a white matter. The gray matter are the synapse-rich areas where actual synapses take place. The brain activity takes place. That's the cortex, the gray matter areas. So this is basic difference between the two.
Okay.
Two different.
Okay. At least grossly we could feel what
Yeah.
differences is. Okay, participants. Any questions? I think they want to have reading material before asking
No,
questions.
that's okay. We see idea is here in such a short course. It is only they should be able to appreciate what MRI can do.
Right.
Yeah. So baki though it will take time for them to grasp the subject. But at least
To read the
they should know what applications are possible using MRI.
So,
Yeah.
sir, practically what I feel as a layman, this MRI can map anything, whatever we think, even.
Yeah. Actually, a lot of work is going on into fMRI and other areas. it's a very, very intriguing.
Very versatile technique looks like.
Yeah, very versatile. Very versatile.
Professor Khusu, see, the thing is that now you said that when you are, let us say, recording the imaging, the gray matter of 20 healthy person, and you saw some difference.
Yeah.
So what it tells actually, that difference means.
See, the idea is, suppose you have a person, okay? You find, when you do a psychological battery, you find his, some of the functions are impaired. They are not as, like response time is very slow, okay?
Hmm.
And then, he is not able to put attention to any work, right? So now
Yes.
you know by subjective methods that he is not able to do such jobs properly. So this gives you a idea where in brain things are happening wrong.
Okay.
This is giving you a diagnostic information, and then after you give therapy, whether the person is, because sometimes people may show their inabilities for some something in their mind, knowingly they are not doing things properly. Here, once you do this, you know sure whether there's a problem in the brain or they are just trying to make up. And here you can do a prospective study that whether the therapies have really helped them or not. And if the therapy is going in the right direction, then you can continue the therapy. Otherwise, you can stop. This most of these cases, these chemotherapy patients, when they were given chemotherapy and anti-cancer therapies, a lot of times they were given therapies, but it was not helping the patients. Then there is a technique called MR spectroscopy in P31 spectroscopy, where they could see whether the tumor is really regressing or not by giving this therapy. And if it is regressing, then probably they will continue the therapy. If it is not affecting the patient, helping the patient in any way, why to give therapy all along?
Yeah. Sir, another question. See, this you are telling the Brahmi,
Yeah.
this Brahmi. So I think in this Indian Ayurveda, it is well known that Brahmi increases the memory power. So
Yeah.
can the memory power can be, let us say, qualitatively diagnosed by this imaging?
Yeah. Actually, sir, problem is, Ayurveda, we all have been saying for last 5,000 years that Brahmi is good for memory.
Yes.
But nobody is, everybody, some people believe and they take it. So, but there's no scientific evidence to show that it is helping.
Okay.
Okay. So by doing this work, by doing functional MRI, showing the results on images, quantifying the data, you can actually prove that yes, brain is improving. The functions are improving when the
Okay.
person is taking Brahmi.
Okay.
And also there is a lot of debate whether Brahmi extract, Brahmi in purest form or Brahmi in desi ghee,
Okay.
which is called Brahmi Grita.
Mm-hmm.
And now we are trying to show Brahmi Grita gives much better results than Brahmi extract.
Mm-hmm.
Brahmi given in with the ghee,
Mm-hmm.
it gives you better results than Brahmi extract itself.
Mm-hmm.
So these are the studies where you have to show scientific methods, scientific data to show that they are actually working.
Mm-hmm.
So this is a because Ayurveda is there, it has so much of potential. But Ayurveda, nobody helped Ayurveda by robust scientific methods
Yeah.
to prove the point.
Mm-hmm.
So that is why allopathy is getting all funds for research, and they show everything on most modern scientific methods. But Ayurveda में वो कुछ भी नहीं है.
That's true. So MRI can help in studying Ayurveda, whether how it is working or how far it is.
Yeah, anything, any intervention, whether it is Ayurveda, MRI doesn't know whether it is Ayurveda or allopathy, but it shows you changes.
Medicine as well as practice, as you told.
Yeah.
Yoga. How yoga is helping in curing the.
And I will show you when we go to the applications. I will show you in the cortical thickness. You know, the people who do meditation for a long time, who are doing practicing meditation for years, their cortical thickness is as good as 25 years old boy,
Oh,
even if they are 55, 60.
oh, that is the reason maybe this which.
Yeah, yeah, and the people who don't perform, their cortical thickness, you know, cortex shrinks, the gray matter shrinks, so their cortical thickness goes is much smaller compared to people who are doing meditation for last 15, 20, 30 years. Their cortical thickness is maintained.
Sir, in this regard, may I just ask one thing? So you see this thickness is changing with yoga. So what kind of change means, what is the reason behind that?
No, because it's a neurodegeneration because of aging. No,
Yeah, that is there. Matlab, means
your gray matter cells, they are dying.
Okay.
They are dying.
So you mean you are changing blood circulation kind of thing to?
Yeah, because by doing this, you are improving your blood circulation. They are getting proper nutrients. You know, so cells are viable. No?
Okay, so basically maybe your circulation, the nutrients you are
Yeah, yeah,
supplying,
everything. This is what actually it
that's the
does.
way it is helping.
Then also, when you are doing these processes, you are also reducing your mental, you know, this overloading of
Yeah.
information and noise in the brain.
You are reducing the stress.
Random thoughts. You are reducing your stress. You are utilizing your energy in a very efficient way. You're not wasting your nutrients in unnecessarily. No, you're preserving them.
So at least we are getting some logic how it works.
Yeah.
Thank you, sir. Thank you very much. Thank you very much. So, sir, if possible, can you send some reading material for the participants?
Yeah, I will do that.
Yeah.
I will do that.
Okay, sir. Then thank you very much for this talk, and
Thank
maybe
you so much.
and
No,
maybe
We meet on
Wednesday.
Wednesday. Yeah.
Okay. Thank you, sir.
Okay. Thank you so much.
So now you start thinking, how are you going to work? Whether working or not working, at least you can have a proof.
See, certain things, na, actually get. See,
কিন্তু আমি আপনার একটা বলে না আনসিনক্রোনাইজ যে ইয়েটা মোড, ওই মোডটাই মেইনলি রেসপন্স।
yes.
ওই মোডটা কিভাবে কাজ করে সেটা যদি ধরতে পারেন